My special research topic was stomach cancer, and specifically I focus on gastrointestinal stromal tumors. Stomach cancer is also known as gastric cancer, which can affect any part of the stomach. In most of the world, stomach cancers form in the main part of the stomach, which is called the stomach body. Stomach cancer is an abnormal growth of cells in the stomach, which you can see in the figure that I have on the slide. And in addition to that, there are different types of stomach cancers, like adenocarcinomas, lymphoma, gastrointestinal stromal tumors, which are called GISTs, and carcinoid tumors. GISTs are what I'm going to be focusing out throughout this PowerPoint. And GISTs are soft tissue sarcomas that can be located in any part of the digestive system, and they're mostly found in the stomach and the small intestine. GISTs start in early forms of special cells of the wall and the GI tract, which are called the interstitial cells of Kegel, which are referred to as ICCs. And sometimes they're referred to as pacemakers of the GI tract because they signal the muscles of the GI tract to contract and move food and liquid, which, helps the digestive system. More than half of GISTs start in the stomach. Most others start in the small intestine. And GISTs start in specialized nerve cells, which are located in the walls of the digestive system. So overall, GISTs are a rare type of stomach cancer, but they can become very harmful in the end. In the United States, GISTs and stomach cancer is more likely to affect the area which is the esophagus, called the gastroesophageal junction. In addition, when it comes to ages, GISTs develop in people of all ages, but they're mostly common between the age 50 and 70, and they rarely occur before the age of 40. GISTs are more common in men than in women, and statistically about 16,000 cases were related to men and about 10,000 related to women. Statistically, GISTs are rare. They make up less than 1% of all gastrointestinal tumors. And each year, about 4,000 to 6,000 adults in the U.S. are diagnosed with the GISTs. Their survival rate is called a 5-year survival rate, and it tells that people live 5 or more years after the cancer is found. And the survival rate overall is about 83%. So basically, if you're diagnosed with this cancer, you have a higher survival rate. In addition, the annual incident report shows that 10 to 20 per 1 million cases are GISTs of stomach cancer, and they arise anywhere in the walls of the gastrointestinal tract, but statistically about 55% are from the stomach, 29% from the small intestine, 3% from the colon and rectum, and 0.5% from the esophagus, which is a less common site for the disease. Lastly, risk factors include diet, nutrition, physical activity. If you're obese, you have a high diet in salty and smoked foods, a diet in low fruits and vegetables, this is more likely to cause GISTs. And if you have family history of stomach cancer, that's also something to be aware of. If you have reflux disease or any stomach inflammation, smoking also is a factor that can affect stomach cancer. And a recent study showed that the risk of cancer is low in the lower stomach, increased by 18% per every ounce of processed meat eaten per day. Lastly, GISTs are less common in the U.S. and they're more common in East Asia. Stomach cancer and GISTs have very common symptoms, and these symptoms are being anemic, abdominal pain. You can also physically feel a growth in your abdomen. If you have nausea or you're vomiting, you have loss of appetite, and when you eat, you feel full after small portions. This will then aid in the weight loss and also malnutrition as well as indigestion. In addition, sometimes there's difficulty swallowing. You can have heartburn, stomach pain, and cramping. And when you need to see a doctor is when you have bloody stool, diarrhea, you're excessively losing weight. So these will bring you to the doctor, and the doctor will then run diagnosis tests. When it comes to diagnosing GISTs and stomach cancer, doctors will first do a CT scan, and this is where you swallow a liquid that makes your stomach and small intestine more visible on x-rays. The scanner takes multiple x-rays, and it will produce a cross-sectional image of your abdominal organs, and then this will show the size and position of the tumor. They'll use a CT scan and determine if an upper endoscopy, which is where you take a tube down your esophagus and it takes pictures and looks at your inner lining of your stomach and your intestines. And then they'll use a biopsy, which is where they take samples of tissues where there is GIST formation or any other type of inflammation. And then they will bring this to a laboratory and run laboratory tests. And this will provide information on the tumor-causing proteins. And the tumor-causing proteins will then allow for tests to be run, like immunohistochemistry, which detects proteins that are controlled by genes of the GIST cells. And this will then help guide treatment decisions. Lastly, determining the cancer severity. So the lowest stage, which is stage 0 or stage 1, is when there's a small cancer and it's just in the inner layer of the stomach and it hasn't spread. But the highest stage, which is stage 4, is when the cancer becomes advanced and it spreads. And this will allow for determination of the treatment. Specific genes to GISTs are the KIT gene. The KIT gene provides instructions for making a member of the protein family, tyrosine kinase receptors. This will transmit signals from the cell surface to the cell and aid in signal transduction. The PDGFRA is a tumor promoter, and it provides instructions for making a protein, which is the platelet-derived growth factor receptor, and this is part of the tyrosine kinases, and this works with the KIT gene. The NF1 gene we also went over in class, and this produces a protein called neurofibromin, and it helps regulate cell growth. So when it's mutated, the gene causes a loss, which allows cells to grow uncontrollably, which causes cell proliferation. The KRAS gene provides instructions for making a protein in the RAS-MAPK pathway, and it signals from outside the cell to the cell's nucleus, and it instructs the cell to grow and to divide. The BRAF provides instructions for making a protein to help transmit chemical signals from outside the cell to the cell's nucleus, and it works with the KRAS in the RAS-MAP pathway. The SRC gene is a non-receptor protein, part of the tyrosine kinase family, and it's involved in controlling cellular processes like proliferation and differentiation. And a small study was done where GISTs have increased of SRC, but unlike in other tumors, SRC may be associated with longer survival. So elevated SRC activities in GISTs improve the immune response to the specific treatment, so that SRC may be downregulated during the treatment therapy. KIT genes and PDGFRA tyrosine kinase genes are pivotal in the development of GISTs because they initiate the oncogenic events. And this happens, as you can see in the diagram, the KIT and PDGFRA receptors bind to the ligand. And once they bind to the ligand, they activate PI3K, which will phosphorylate PIP2 and PIP3, which you can see in the diagram. And this diagram also shows that P10, which is a tumor suppressor gene, will regulate and balance between PIP2 and PIP3. This will then activate AKT, which will be phosphorylated, and it will activate the BAD protein, which inhibits apoptosis, and mTOR, which stimulates protein synthesis, which aids in cell growth. So this will cause cell proliferation, which is when the GISTs will form. When it comes to mutations in the KIT and PDGFA genes, this leads to proteins that are no longer require the ligand binding to be activated. So the proteins and the signaling pathways are turned on, which increases the proliferation and survival of cells. So when these mutations occur in the ICCs, which I talked about earlier, which are in the lining of the smaller intestine and in the stomach, this allows for uncontrolled cell growth, which leads for the GIST formations. These mutations in the KIT and PDGFRA genes are associated with familial and sporadic GISTs. This is the RAS signaling pathway, and we know the RAS protein is activated when binding to GTP and inactivated when binding to GDP. So, as you can see at the top, FR are growth factor receptors that binds to a ligand and activates the SOS and GRBT, which decide to turn RAS on or off. EGFR is involved with overexpression and truncation. Overexpression involves multiple gene copies and makes too many mRNAs, and truncation deregulates protein activity and which will then break fragile DNA. So the SOS and GRBT will decide to turn on the RAS gene, which you can see, which then trigger RAF. And BRAF is actually the mutated version of RAF protein. So once RAS is activated, it turns on early genes like JUN and FOS, and this will then cause cell proliferation. It's also common to find mutated RAS protein in tumors where G-alpha is active without a signal. Lastly, it is important to note that mutations in the RAS signaling pathway genes, like the KRAS and BRAF, are also reported in the KIT PDGFA wild-type GISTs, and shows that this is a rare mutation of the RAS signaling pathway. Doctors take in consideration many different factors to determine treatment options. TNM stands for tumor node metastasis, and this is a staging tool that is common in most cancers, but it's not required to stage GIST. Instead, they're going to look at different factors of how aggressive it is. And the most common way for treatment of GISTs that haven't spread is surgery. Now, looking at the diagram in A, you can see the two arrows are pointing to the GIST that is going to begin to spread. So what they do is, this is a treatment called amitabin, which I'll go into next on the next slide about treated drug therapy. But this is just a picture showing that after 8 weeks and then after 12 weeks, and then on the C picture, you can see that there is no metastasis or any GIST, and this is after surgery and after treatment. Drug therapy is another common way to treat GISTs and stomach cancer. Targeted therapy is when you are identifying the pathways and the activating mutations like BRAF, which is in the RAS signaling pathway, as well as the KIT and PDGFRA inhibitors. So most metastatic GISTs respond to therapies that have these inhibitors in them. The targeted drug therapy focuses on abnormalities present within the cancer cells. It blocks these by targeting drug treatments that cause cancer cells to die. So for GISTs, the target for these drugs is the tyrosine kinase receptors that helps cancer cells grow, and which is cell proliferation. Secondly, amitabin, which is the chemical composition in the left lower part of the slide, the picture. This is the first-line targeted therapy used to prevent GIST recurrence after surgery. This drug is used to shrink the tumor before surgery, and then it's used after, as long as the drug is still effective and it controls recurrent GISTs. So this proves that it is a selective tyrosine kinase inhibitor that is used for standard care of GISTs. As you can see in the picture, before treatment, this is before surgery, and before treatment, all the cells in the nucleus are taking up the cytoplasm, and as you can see, there's a lot of them. And then after treatment, after surgery, this is what the cells look like. They're more spread out. Overall, treated drug therapy is amitabin is the most popular way to treat GISTs.